I want to jump right into glaucoma. So as you know, in glaucoma, this is a disease characterized by anatomical changes to the optic disc, nerve fiber layer, and the irreversible loss of vision that's associated with that. And as I'm sure you're also aware, that the compliance with topical drops is very well understood to be pretty bad, horrible in fact, and that lack of adherence leads to disease progression. So there have been some recent advances with surgeries and some of these drainage devices. But these are really not good options for all patients. I think the long-term outcomes are to be determined. Some good progress, but not for everyone. And when you look at the extended release drug systems, again, some creative approaches here, but in our view, not really hitting all the technical targets in terms of duration and full efficacy. So our targets are here. Basically, we want to get full prostaglandin efficacy 24/7, greater than 6 months duration from a single dose, easy administration, of course, less hyperemia than topical drops, fully biodegradable, and excellent safety. And we achieved that by using a core technology called PRINT, and it's outlined here as what you see on a silicon wafer. Anything you can do on a silicon wafer, we can use to design very precise drug products. So PRINT is, in essence, essentially a particle molding technology. It's licensed from Liquidia, who's using it in multiple therapeutic areas. And think about it as where nanotechnology meets formulation science. We have the ability to make really precise and reproducible tiny dosage forms that can work in many different applications. And as exotic as it is, we've already transferred this technology to a commercial supplier, and we're on our way to hitting near commercial specs early in development. So in essence, what we have here is an intracameral, fully biodegradable travoprost that goes in the anterior chamber and delivers drug right next to target tissue. We have a custom injector, allows us to do that very easily for a very simple in-the-chair procedure. And last year, we presented some early results from our first cohort where we showed the high-dose ENV515 was numerically equivalent in IOP lowering to Travatan Z in terms of day 25 diurnals. And the low dose was about like a timolol-like effect at about a 20% change from baseline. Obviously, we like the high-dose efficacy, and we moved forward to manufacture some new material, but while we were waiting for that new material to be made, we decided to take the low dose into a cohort where we could follow the patients for 12 months and get a read on the full duration of action of this formulation. So be patient with me as I walk you through this cohort here. So I'm going to show you the data from a cohort we started in a handful of glaucoma patients. They came in as prostaglandin responders on prostaglandins once a day. They came in about 20. In this case, we're using timolol as our contralateral control eye. They wash out. You can see the baseline diurnals here. And then their pre-dose 8 a.m. IOPs are about 26. So that's their fully washed out IOP. And now you can look at the diurnals at weeks 2, 6, and 12. And as you recall, these are the key time points. They're typically used for assessing non-inferiority in large pivotal trials. And you can see that ENV515 in blue versus timolol in yellow are very well matched here. And in fact, when you look at where they started on a once-a-day prostaglandin in the wild, we're at or below where they were on standard of care. So great results so far. We now have results for months 4, 5, and 6. And so that's now getting to our target product profile of 6 months or more. And you can see, again, the IOPs are very well controlled. Again, single dose, IOPs controlled 24/7. And now we're excited to show you that we also have data out to month 7, 8, and 9. Again, IOPs controlled, single dose, 9 months and going. We expect attenuation over the next few months as we get to months 11 and 12. And at that point, the polymer should be fully dissolved and ready for redosing. So very excited about the results to date, but we know we can do better. But first, let me review the safety. So we see some minor procedure-related AEs, typically some foreign body sensation, a little bit of hyperemia. That comes back to baseline fairly quickly. Importantly, no changes to the cornea, so no changes in endothelial cell counts or corneal thickness, and the hyperemia is pretty much unchanged at steady state. So hitting our profile there as well. And this gonioscopy picture is just a reminder that we can watch these over time. We can watch them monthly, and you can see the eye is quiet, and these are nestled nicely in the angle. So the opportunity here is to get it to the next level. We've got a high dose coming online, again, repeating what we saw in the first cohort, and to let these guys go on for 12 months. That cohort is starting now. and we're expecting to see full prostaglandin efficacy, again, greater than 6 months now, probably greater than 9 months of efficacy from a single dose. And we've taken this new formulation into our dog model as well, and we already see 40% change from baseline for 100 days and counting. So excited to bring this forward in our dose escalation strategy in the clinic, and we believe this is a really big commercial opportunity, $1 billion plus. Here's our portfolio in a nutshell. So you can see where we are in glaucoma. We're going to have a large, well-controlled trial that'll start in the second quarter of 17. They'll read out in mid-18. And some other portfolio programs here. We have a ENV1105 for diabetic macular edema. That'll be in the clinic later next year. And also a wet AMD program where we're trying to extend the duration of action of some of these anti-VEGF agents. And I'll talk to you about that in a second. This portfolio is funded by a Series A that was led by Canaan and NEA, with Pappas and Morningside joining that syndicate. So a little bit on 1105 here. So this is essentially a fully biodegradable intravitreal dexamethasone. It's designed to be 6 months or greater in terms of duration of action. And it's a very linear formulation. So really what we're trying to achieve here is consistent tissue levels in the back of the eye for 6 months or more, and to do that in a very efficient way so that we can be very dose sparing and even get away with a smaller needle. So we're excited to bring that into the clinic later next year. The other area of research that we're working on is trying to get these anti-VEGFs to last longer. It's been a tough problem to solve, but we have some interesting results in a primate model where we've done a pilot PK study. So you can see Eylea in yellow injected on label and the vitreous levels, and then our formulation of aflibercept here with 3 months of similar or even higher vitreous levels from a single dose. And so a really nice proof of concept. We basically feel like we're in the right spot in terms of payload and drug release. So finally, hopefully you got a good sense of what we're doing at Envisia and the great progress we made in glaucoma. And your moment of zen here are a few SEMs of some new formulations we've done with about a half a dozen TKIs. Thank you.